Every experiment begins with a plan. Before you plant a single seed, before you run a single assay, you need to decide how many plots, which seeds go where, and what counts as a fair comparison. In computational biology, this means sample size, randomization, and controls. Get the design wrong, and no amount of statistics can save you. Welcome to the garden planner. How many tomato plants do you need? Plant 3, and one bad seed ruins your experiment. You can't tell if the fertilizer failed or if that one plant was just unlucky. Plant 30, and the bad seed is noise, not signal. Sample size determines whether your experiment has the power to detect a real difference. Too few samples, you miss real effects. Too many, you waste time and money. The sweet spot comes from a power calculation, a question we'll answer in part 2. You have 20 plots. 10 get fertilizer, 10 get water only. If you put all the fertilizer plots near the fence where there's wind shelter, you can't tell if better growth came from the fertilizer or from the protection. Randomization breaks this link. Shuffle which plots get which treatment. Now wind shelter, soil quality, and sunlight are evenly distributed across both groups on average. This is Fisher's insight from 1935. Randomization is the foundation of causal inference. Without it, you have correlation, not causation. Some plots get morning sun, others get afternoon sun. This isn't something you can randomize away, it's a known source of variation. So you block on it. Within each sunlight block, randomly assign fertilizer and control. Now you're comparing fertilizer versus control under the same lighting conditions. Blocking reduces noise and increases power. In biology, this is the paired design. Same patient at 2 time points, same litter of mice split into treatment and control. The block absorbs the between-subject variation. The plot with no fertilizer. Without it, you don't know if your tomatoes grew because of the fertilizer or because it was a good year for tomatoes. The negative control water only sets the baseline. The positive control, a known effective fertilizer, confirms your assay is working. If the positive control fails, the experiment is invalid regardless of the treatment result. In drug development, the vehicle control and the standard of care arm serve exactly this purpose. 4 principles before planting. Sample size, enough plots to detect the effect. Randomization, shuffle to break confounders. Blocking, group by known nuisance variables. Controls, baseline and positive reference. Get these right, and your data is interpretable. Get them wrong, and no statistical method can fix it. Next, advanced layouts for complex questions.